graduate school and studies the transcriptional regulation of blood cell development. So turn it over to Lou.
Thank you. Sorry to delay lunch a little bit. I'll try to be quick. So I'm going to talk a little bit more about DNA manipulation and measurement to sort of add to what Bento already told you about, and I'm also going to talk about manipulation of RNA levels and measurement of that. Bento alluded a few times to the central dogma of molecular biology. This is the sort of understanding that DNA can replicate itself. It can also be transcribed to RNA, and then RNA is translated to protein. And so the idea is that information flows from DNA to protein and not in the other direction, which we know to be untrue in some cases now. So proteins are the ultimate effectors of phenotype, and so proteins like hemoglobin, which carries oxygen in your blood, and then other proteins are involved in connecting cells together. So basically everything that happens is the result of it, or just about everything that happens is the result ultimately of a protein exerting its function. And so like I said, there are also special cases that we know of now where RNA can replicate itself, and RNA can also be reverse transcribed from RNA to DNA, and I'll discuss that a little bit more later. But what I'm going to focus on here is manipulating DNA and RNA, which you can see would have an obvious effect on protein levels ultimately. So there are two major approaches to molecular genetics. One of them is what we now call forward genetics, although when it first started, I imagine it was just called genetics. So this is to identify, this is a method that's used to identify genes that are responsible for observed phenotypes, such as taking a bunch of worms and inducing mutations, and then going back from that and seeing how how a certain phenotype was arrived at. So if you mutate a bunch of worms and some of them can't move, you can then do forward genetics to determine what genes were mutated that caused these worms to not be able to move. A sort of a more recent approach now is the approach of reverse genetics, and that's what I'm going to talk about, where we can modify or manipulate DNA and RNA levels and so perturb expression levels of single genes or multiple genes in a screening sort of environment. And then we can determine the function of those genes by studying the effect of that perturbation on the phenotype. So basically we can overexpress or knock down genes, and based on what happens to the cells or the organism in question, we can infer function of those genes. So like I said, I'm going to talk about mainly DNA and RNA. So I'll briefly touch on cloning and polymerase chain reaction, which Bento already talked about, and a little bit about delivery of DNA into cells, which he went into also. And then as far as RNA, I'll talk about reverse transcription-based PCR and also real-time PCR, which are methods we use to measure RNA levels, gene expression profiling, which we use to measure global RNA levels, and gene knockdown, which is a method of perturbing RNA levels to study gene function. So this is a view from a publicly available genome browser website called the University of California, Santa Cruz Genome Browser. So this is just about a 200 kilobase region of chromosome 18 in the mouse, and it's you know, you can see there's a fair number of genes here. These are all the protocadherin beta genes. That's not really important. I just wanted to show sort of a view of a bunch of genes, and if say we wanted to clone protocadherin beta 8, how we might go about doing that. I picked this region not because I'm particularly interested in this specific gene, but because it's sort of an easy case, as these are all single exon genes. So we don't have to, well, I'll get into that a little bit later. So if we just zoom in on the area right at the beginning of this gene, we can see the DNA sequence here, and underneath that is a translation of that sequence into a protein sequence. So this region upstream here of the gene is not normally translated. The genome browser doesn't care about that. It turns every triplet into an amino acid. But so the gene starts with an ATG, which is translated into a methionine, and then goes on from there. And if we zoom in then on the sort of the back end of the gene, we can see that it ends with a TAG, which is one of the stop codons. And so this gene is just transcribed and then translated from the DNA to the protein along this entire length, and stops at the end when it reaches the stop codon. And so if we were to clone this, this is the region we'd want to clone, from the ATG to this TAG. And so if we just look at, here's our ATG again, here's our TAG, and then there's all this sequence in the middle, which we don't even have to know in order to clone this gene. So I'll talk a little bit about designing primers to sort of to set up the boundaries for the region of amplification during polymerase chain reaction. So if we look at the DNA in a more accurate representation, it's actually a double-stranded molecule. And so on the top, we're looking at a 5' to 3' look. So each of these bases there's a directionality to them. And so they're arranged in this anti-parallel sense. And so what we want to do is amplify both strands of this to make a copy of this gene. And so if we imagine that we want to amplify from this ATG to this TAG, we would want to design primers that are complementary to these regions. I'm sorry, that are composed of these regions that are highlighted in yellow. And that'll become ultimately our PCR amplicon. So Bento's movie about polymerase chain reaction did a very good job explaining how this works, and I'll just go through it very quickly. The two parental strands are melted at 95 degrees, and then the primers are actually in this mix already, and when the temperature is lowered, these primers are able to anneal. So these are these short sequences in yellow. So this one would anneal to the bottom strand, and this sequence in yellow would anneal to the top strand. And then when we raise the temperature to 72 degrees, the polymerase will extend along these. And as we cycle through this 25, 30, 35 times, we get an amplification of the region between the forward primer and the backward primer. And you can see there's an exponential growth of the or exponential accumulation of the regions that we want. There's a linear accumulation of these sort of hybrid molecules, and the parental strand is not amplified at all. So you wind up with a population of DNA that is greatly enriched for these shorter pieces that are bounded by the primers. And if we want to use these sequences for cloning, one of the tricks that we can use is on the back end of these primers. So on the line part of this arrowhead, we can add recognition sites for restriction enzymes, so that after we're done amplifying this, we can cut the end of these so that we have a staggered end, like Bento had shown before, that we can then ligate into a vector. And so if we imagine that this is our PCR amplicon and we've put restriction enzyme sites on the end of it, we can now cut this with a restriction enzyme, either the same one on both sides or different ones, and get this sort of staggered piece with overhangs that can then be ligated into a vector that is also cut specifically with the same enzymes. And then we can take this now circular vector, so this actually is a complete circle with just a piece cut out of here. We can take this circular vector, transform it into bacteria, and then amplify and further amplify that and purify this DNA piece out of the bacteria. At that point, we can take our purified DNA vectors and deliver them into a cell in a number of different ways. Bento already discussed electroporation and liposuction, and I think he touched on microinjection also, and mentioned viral transduction. So there are obvious benefits and drawbacks to each of these methods. I'm not going to get into the specifics of all these because everybody's probably hungry. So reverse transcription PCR is the way that we would normally clone a gene out of the genome. So if we were to try to clone this GATA2 gene the same way that I just showed how we cloned that protocadherin gene, we would wind up with we'd start with a primer on this end and a primer on this end, and when we want to amplify this out of genomic DNA, we'd wind up with a huge piece of DNA that contains all these intronic sequences that don't actually code for the protein sequence. So while these aren't necessarily noise or junk DNA, in the context of cloning a gene, we don't need all these pieces, and actually they make it harder to do downstream applications because we wind up having all this DNA in there that isn't doing anything functionally for us in general. And so that makes it more difficult to make viruses and more difficult to transfect the cells because we wind up having larger pieces of DNA. So during normal transcription, all of this area is transcribed, and then these intronic regions are spliced out, and that'll make a single-stranded mRNA that is composed of only these big blocks of blue, which I've shown here, which is then or polyadenylated. So at the end of transcription, just a long string of A's is added to the end of the RNA, and that's sort of it's something that the cell uses to identify degraded RNAs, as degraded RNAs won't have all this poly-A. So we can take advantage of this and design primers that are long strings of T's and add polymerase to this, and we'll be able to reverse transcribe this mRNA and now get the opposite or the reverse complement sequence of this mRNA, which we call cDNA or complementary DNA. We can take this cDNA and use that as our template for PCR and amplify just the coding region of the gene, and use that to transfect cells or make viruses. So now I'll talk a little bit about a method that we use to detect the levels of RNA in a cell for a specific RNA. So if we have one specific RNA that we're interested in knowing the levels of, we can design a probe. So these are all these colorful lines here are supposed to be DNA bases, and then we have a fluorescent reporter and a quencher that are attached to either side of this probe. And so in the context of this probe, this reporter is actually quenched by fluorescence resonance energy transfer. This reporter's fluorescence is quenched by this quencher. And so when this probe is intact, the only signal you would see is this quencher. So the way this technique works is that you would design two primers, one at either end of of the region that you're looking to detect, and you also design a probe in the middle of this region that you're looking to detect, and it has these this reporter and this quencher on it. And so during the annealing step of PCR, this probe will anneal to the DNA. And then during the extension step, Taq polymerase will come along in the 5' to 3' direction from left to right here on the top, and it will actually chew up this primer that's already there. And this reporter is then released, and it's no longer being quenched by the quencher on the other end of the probe, since they're not in physical proximity anymore. And so then we can measure this in a real-time PCR machine after every cycle, and determine how much of this is being released on every cycle. And that should be directly correlated to how much template we started with. All right, so another method that you probably will hear a lot about is microRNAs, or global gene expression profiling. And so this method, again, relies on hybridization between complementary strands of nucleic acid. And so if we're looking at sample A, maybe this is a treatment of our cells, and sample B, which is our untreated cells. And a treatment can be anything, a drug treatment, a gene knockdown, a gene overexpression. And so we want to basically look then at the overall gene expression profile and the differences in all of the genes between sample A and sample B. So what we can do is do this reverse transcription on all of the RNA that's isolated from the cells, and label that cDNA with for instance, a green dye. And over here, we would do the reverse transcription and label the cDNA with a red dye. When we mix these together and hybridize them to a chip that has DNA oligos spotted on it, we can then detect how much of this cDNA is hybridizing to the DNA that's spotted on the chip. And so there are 3 scenarios basically where we have, well, there are 4 scenarios. One is that there is no RNA that hybridizes to a particular probe, in which case it would be completely black, which you can see a number of on this chip. The next is that there is a lot more of sample A than sample B, in which case it would be green. A lot more of sample B would make it red, and relatively equal levels of red and green will read out as yellow. And then the last thing I want to talk about, so we've talked about gene knockdown a few times, but not with any sort of specific mention of what it is. So the idea here is that, like I was saying, we like to perturb gene expression to use that to infer gene function. And so one of the more recent advances is the ability to specifically knock down the levels of particular mRNAs within the cell. And so we can use these, so we have siRNA gene silencers, which are short double-stranded RNAs, shRNA plasmids, which are circular pieces of DNA that contain a region that will be transcribed into an RNA that can be processed by normal cellular processing machinery. The shRNA lentiviral particles are fairly similar to the plasmid DNA, except that this DNA is going to actually incorporate into the genome, which has its benefits in some situations. And so this shRNA hairpin here can be processed by the siRNA processing machinery in the cell, namely Dicer and Drosha. And then it's loaded into this RNA-induced silencing complex, where one strand is discarded and one strand is loaded into this complex, and you have this activated RISC complex, and then this activated complex can associate with an mRNA, again in a hybridization-dependent manner, so in a manner that takes advantage of the complementarity of the RNA with another strand of RNA. And so when this mRNA that's being targeted hybridizes with this siRNA that's loaded in the RISC complex is able to cleave the target mRNA and destroy it, which leads to obviously a lower amount of protein being produced by this particular species of RNA. That's all I've got, and I'm happy to take any questions. Yeah. It's coming. Now that we've finally learned to wait for it.
I was just wondering, what are some of the drawbacks to using electroporation for getting DNA into the cells?
There's a fairly high rate of cell death when you shock them, which is not so surprising. But if you have low numbers of cells or very sensitive cells, and also different cell types sort of take differently to the different transfection methods. So some cell types are a little bit more resilient to the electroporation than others are. But if you have low cell numbers, you can't really deal with losing 50% of them in some cases. So that's one of the drawbacks.